Audio Jungle.
Hi guys, welcome to my video, and in this video we're going to discuss the cellular respiration and fermentation. In general, a cell contains only enough ATP to sustain from 30 seconds to a few minutes of normal activity. Because it has such potential energy, ATP is unstable and is not stored. As a result, most cells are making ATP all the time. This video discusses the production of ATP through cellular respiration and fermentation. You can think of the complete oxidation of glucose via cellular respiration as a set of 4 interconnected processes that together convert the chemical energy in glucose to chemical energy in ATP. Each of the 4 processes consists of a distinctive starting molecule, a series of chemical reactions, and a characteristic set of products. So the first process, an aerobic process, is the glycolysis, which happens in cytoplasm and not in the mitochondria. During glycolysis, 1,6-carbon molecule of glucose is broken into 2 molecules of 3-carbon compound pyruvate. During this process, ATP is produced from ADP and P and nicotinamide adenine dinucleotide, the NAD, and it is reduced to form NAD. The second process is the pyruvate processing. In pyruvate processing, pyruvate is processed to release 1 molecule of carbon dioxide, the CO2, that we breathe out, and the remaining 2 carbons are used to form the compound acetyl-CoA. The oxidation of pyruvate results in 1 more NAD, and again it will reduce to a NAD. The third cycle is the citric acid cycle. Each acetyl coenzyme is oxidized to 2 molecules of carbon dioxide. During the sequence of reactions, more ATP and NAD are produced, and flavin adenine dinucleotide, or the FAD, is reduced to form FAD. So, in the Krebs cycle, it produced 2 ATP at the end of the process. In an electron transport and oxidative phosphorylation, electrons from NAD and FAD have higher electrons and bring to the last step and move through a series of proteins that together are called an electron transport chain, or the ETC. The energy released in this chain of redox reaction is used to create a proton gradient across a membrane. The ensuing flow of protons back across the membrane is caused to make ATP. Because this mode of ATP production links the phosphorylation of ADP with the oxidation of NAD and FAD, it is called oxidative phosphorylation. So let's see it closer. So in this diagram, the electron of FAD and NAD move through a series of proteins. Every time the electron passes through this protein, it will pump up proton ions out, causing to have a tons of positive ions in outside of the matrix. Then what will happen is that the proton that pumps up from the NAD and FAD will move through the protein called ATP synthase and will be the site of the ATP synthesis. Every protons that comes through it, we're going to generate ATP by attaching this ADP to it to produce ATP. In an electron transport chain, we are making a lot of ATP, just 32 ATP compared to the past processes. So let's move on to anaerobic fermentation. In fermentation is a metabolic pathway that regenerates NAD by oxidizing stockpiles of NAD. The electrons moved from NAD are transferred to pyruvate, or a molecule derived from pyruvate, instead of an electron transport chain. Cellular respiration and fermentation are alternative pathways for producing ATP. When oxygen, or another final electron receptor used by the electron transport chain, is present in a cell, the pyruvate produced by glycolysis enters the citric acid cycle, and the electron transport system is active. But if no electron receptor is available to keep the ETC, or the electron transport chain, running, then pyruvate undergoes process known as fermentation. When you run up a long flight of stairs, your muscles begin metabolizing glucose so fast that the supply of oxygen is rapidly used by their mitochondria. When oxygen runs out, the electron transport chains shut down, and NAD cannot donate its electron there. When fermentation takes place in your cells, the pyruvate produced by glycolysis then begins to accept electrons from NAD. This process, called lactic acid fermentation, regenerates NAD by forming lactate. Lactic acid fermentation, the protonate form of lactic acid. The problem is this: lactate acid is very dangerous to our health and have to break down, and it takes oxygen, and you have to breathe heavily after running the long stairs. While the alcoholic fermentation, which occurs in the eukaryote such as Saccharomyces cerevisiae, strains of which are used to make bakers and brewers yeast. When yeast cells grows in bread, dough, or a bottle of grape juice, they quickly use up all the available oxygen. Instead of using NAD to reduce pyruvate, yeast first convert pyruvate to 2-carbon compound acetaldehyde. This reaction give off carbon dioxide, which causes bread to rise and produces the bubbles in champagne and beer, or so-called carbonation, known as carbonation. Acetaldehyde then accepts electrons from NAD, forming the NAD required to keep glycolysis going. The addition of electrons to acetaldehyde forms ethanol as a waste product. The yeast cells excrete ethanol as waste. In essence, the active ingredient in alcoholic beverages is like yeast urine. Cells that employ other types of fermentation are used commercially in production of soy sauce, tofu, yogurt, cheese, vinegar, and other products. The products of those reactions are responsible for many of the complex flavors in these foods. Bacteria and archaea that rely exclusively on fermentation are called oblate anaerobes. So, cellular respiration and fermentation is doing by humans, plants, animals doing it, and even bacteria. So, what exactly is the ATP? ATP is the main source of energy for most cellular processes. The building blocks of ATP are carbon, nitrogen, hydrogen, oxygen, and phosphorus. Because of the presence of unstable, high energy bands in ATP, it is readily hydrolyzed in reactions to release a large amount of energy. Functions of ATP in cells in our body are: it acts as active transport, it acts as cell signaling, structural maintenance, and muscles contraction, and it helps synthesis of DNA and RNA. Even as evolutionists wonder about life before the complex ATP molecule and alternatives to ATP, no other energy source currently exist that can accurately respond to the energy needs of the cell and carry out its crucial processes. That's how important the ATP in our body. So I think that's it. I hope it's helpful.